Welcome to Chapter 10. The subject or the topic of this time is cell reproduction. This is the first lecture of 2. I love this image, this micrograph of cells in various stages of reproduction or division and various stages of mitosis, which is the subject of this chapter. And you can see, for instance, this cell, the dark purple little stringy things that you see in there are chromosomes. And yes, you can see these. Once they get to this point, you can see them with a simple light microscope. But you can see the chromosomes here, kind of lining up in the middle. We'll see that process as we go into mitosis. Here the chromosomes are dividing or separating. You have a group separating to this side and a group separating to that side, and these will become the nuclei of your two new daughter cells. And here you can see a cell. It's one cell that has divided into twos. We have a double division going on here. And the chromosomes are being separated, so each one of these halves will divide in half again. So you'll end up with 4 identical cells. And then you see some cells over here that have recently divided. See how small the nuclei are and how close they are together, so you can tell that those have recently divided. So we'll look at all these stages in this chapter. So one of the characteristics of living things is the ability to grow and reproduce and pass on genetic information to the next generation. And that is what this chapter is all about. The process is called mitosis for higher organisms, plants and animals, and we'll look at each of the steps along the way and see how that happens. All living organisms, including bacteria, butterflies, pine trees, petunias, lions and tigers and bears, oh my, and yes, even humans grow and reproduce. Cell division is absolutely necessary for growth and development. You get a cut on your hand, wound healing, that's cell division. Replace cells lost through normal everyday wear and tear. You take a shower, you slough off some skin cells. Those skin cells have to be replaced. Cell division. Division of one cell into 2. We call the cell that is dividing the parent cell. And we call the cells that result from the division, we call them daughter cells. It's a simple process for prokaryotes. It's called binary fission, and you end up with clones in that respect. But obviously it's more complex for higher organisms like you and I. So your objectives for this chapter, this is chapter 10, describe the process of binary fission. That's cell division for bacteria or unicellular organisms. Describe the role of condensing and cohesion proteins. Distinguish between homologs and sister chromatids. Describe eukaryotic cell cycle. We call that cycle mitosis. Differentiate the events of the S phase. S stands for synthesis. Describe the phases of mitosis. Differentiate cytokinesis in plants and animals. A little bit different. Explain the importance of checkpoints in the cell cycle and explain the role of p53 in cancer formation. Okay. So your book begins with eukaryotic cell division, and at the end of the chapter it goes into binary fission of prokaryotes and unicellular organisms. I'm going to begin here with cell division of prokaryotes or binary fission, which also happens in unicellular eukaryotes, but I'm going to begin here because it's a much simpler process, and that way we can focus intently on eukaryotic cell division. So every living organism needs to reproduce in order to increase their numbers. Remember, propagation of the species is the key in biology. binary fission is asexual reproduction. Now, remember what I said before, anytime you put an A or an AN on the front of the word, it generally means without. So binary fission is asexual. That's reproduction without sex. So there is, in other words, no male or female with regard to bacteria or amoeba or paramecia. So, binary fission occurs with unicellular organisms. There you go. Asexual means without sex. No male or female bacteria. So the genetic material is first replicated, duplicated, we call that replicated, and then it's segregated or separated so that two cells can then form around the new groups of genetic material. Cell division occurs as new cell wall forms. We call it the septum, and separates the daughter cells. These are identical, which means that they are clones. Not so with regard to animals. Generation time for bacteria varies from species to species. E. coli, that's Escherichia coli. E. coli, you're familiar with E. coli from the news, I'm sure. E. coli, model organism for biologists, by the way, is about 30 minutes. Binary fission is exponential, so that every time one cell divides, you get an exponential growth. So if you have one cell divided into two, then those two cells divide into four, those four cells divide into 8, and so on like that. So the population doubles every 30 minutes. Alright, so I've included a video for you on binary fission that's embedded in your in the notes here, so you should be able to access that without going outside to YouTube. So in other words, you should be able to access all these videos through your school computer. But that's a little animation that explains binary fission for you. So binary fission is very simple. We have just a few steps to remember. Replication of DNA, remember that means duplication of DNA. The cells elongate because you got to make room for two new cells. So you have to kind of double in size, if you will. Segregation of the duplicate chromosomes, so you got to get separation of the one group to one side of the cell, the other group to the other side of the cell. Septation. A septum is just a dividing wall. So we have to build a septum. The process is called septation between the two cells, and then the daughter cells separate. Here you see two paramecia in the process of separating. And in the diagram over here, you see the process of replication going on here. There you see, remember that a bacterium has a single circular chromosome. So here you see the two chromosomes. Here you see the chromosome being duplicated. Here you see the two resulting chromosomes, which are clones of each other. There they get separated. Here we have elongation of the cell, where you have one chromosome going to one end, the other chromosome going to the other end. Septation occurs and the two cells pop apart. That's binary fission in a nutshell. The video that I have for you here is a little bit long. You can forward it up to, I forget where the point is, but anyway, you can forward it. But it shows protozoa, paramecia in this case, going through binary fission in real time. So it's pretty cool. I just put that on there for interest's sake. Okay, eukaryotic chromosomes. Now when we talk about mitosis and the cell division of eukaryotic cells, we have to talk about chromosomes. Here you see a nice high-powered electron micrograph of chromosomes, and they generally, I mean, we think of them as taking this X, kind of this X shape. Okay, Chromosomes are thread-like structures located in the nuclei of both plants and animals. We're talking about higher organisms here. Chromosome is made of protein and a single molecule of DNA. So each chromosome is a single long strand of DNA made up of nucleotides, right? So, and by the way, the sugar in DNA is deoxyribose. Okay, so one strand though, it's interesting that you get this sort of this humongous structure here out of a single strand. That's all a single strand. We're talking about one side of the X. It's a single strand of DNA. All right. And just for your information, DNA comes from the Greek meaning color because they strongly stain, and that's why back in the first micrograph that I showed you, you can see the chromosomes really clearly because they take on a lot of stain. So every species has its unique number of chromosomes. Humans have 46. That's 2 pairs of 23. So we have 23 chromosomes, and then we have a duplicate pair of 23 for a total of 46. And those two numbers, 23 and 46, will become important, as you'll see, as we get into mitosis. If an organism has too many or too few chromosomes for that particular organism, so in other words, if humans instead of 46 we have 47, then that causes problems. Or if we have 44, then that causes problems. Generally the problems are fatal, but for example in the case of Down syndrome, Down syndrome results from 3 copies of chromosome 21. It's often referred to as trisomy 21. That's 3 copies of chromosome 21. That results in Down syndrome. Obviously the affected, it's not fatal to the affected, and folks with Down syndrome go on to live perfectly normal lives with some limitations. Okay, as I said in the previous slide, chromosome number varies with species. You can see the typical everyday run-of-the-mill earthworm with its 36 chromosomes. There's cannabis with 20. Saccharomyces cerevisiae, that's a yeast, 16 chromosomes. There is our infamous Drosophila melanogaster, that's fruit fly, with its 8. Here's a dandelion with 4 chromosomes. The most complicated, most advanced species on the planet is Homo sapiens. That's you and I. We have 46, and you'll notice that all these numbers are even. But look at this. Here's Pan troglodytes. That's your typical everyday chimpanzee with 48 chromosomes. So they have 2 more chromosomes than we do. Interestingly, here's this rooster with 78 chromosomes, which is many more than even Homo sapiens. But what I found even more interesting than that was this single-celled organism. This is kind of like a paramecium. It's eukaryotic. 16,000 chromosomes. 16,000. How does that happen? This unicellular, very simple, doesn't have a brain organism, 16,000 chromosomes, and the most complicated advanced species on the planet, Homo sapiens, with only 46. How does that happen? It's interesting, I thought it was interesting, that this organism doubles its chromosome number with every cell division. So every time it divides, its chromosome number doubles. And every time our cells divide, right, we cut them in half. We have them. We go from 46 to 23, right? So that's how we keep our number down. This dude over here has no way of controlling that. Anyway, thought that was interesting. Okay, so we have a quick check here, and the problem is Down syndrome is a result of 3 copies of chromosome, fill in the blank, 21, 23, 46, 12. Well, if you recall that I just said a little while ago that Down syndrome is also known as trisomy 21. That's 3 copies of chromosome 21. Congratulations if you got that right. Alright, so let's take a closer look at chromosomes. Now, homologous chromosomes are pairs of chromosomes. We call them homologs for short. They're homologous. That means they have the same information. Okay, that's what we call them homo. Homo means the same. Homologous chromosomes carry the same information. So here you see 2 homologous chromosomes. Now, it's going to be important to keep the terminology straight. Here you have 1 chromosome, but it has 2 arms. Looks like 4, but this is a short arm. It has 2 sides. This is a short arm and a long arm. So this is one side. This is the other side, and the sides are connected here in the middle at the centromere, it's called. So this is 1 chromosome, okay? Remember I said you have 46 chromosomes. That's 2 pairs of 23. So you have a set of 23 and then an identical set of 23. So here is its mate, its identical twin, if you will. I'll use those terms loosely here because we're just talking about chromosomes. But so this chromosome carries the same information as that chromosome. These are the same genetically. These are homologs, homologous chromosomes. Now each chromosome, as you can see, has 2 sides, and each side is called a chromatid. So the whole thing is called a chromosome. Each side is called a chromatid, and because these again are duplicates of one another, we refer to them as sister chromatids. It has no bearing on gender here, okay? We just call them sister chromatids. So here you have a whole I refer to them as whole chromosomes. Consists of 2 sister chromatids connected at the centromere, at a centromere. And each one of those chromosomes has an identical twin chromosome with the same genetic information made up of 2 chromatids connected at a centromere. Okay, so those are homologous chromosomes. So sister chromatids are 1/2 of a chromosome. Each chromosome composed of 2 sister chromatids joined at the centromere. It's what I just described for you. So then we can start thinking about those numbers, 46 and 23. So one set of, we're talking about humans now, one set of chromosomes, there are 23 of them, 23 unique chromosomes. Now that 23 is 1/2 of the 46, and so for that reason we refer to it as haploid, and we denote it with the lowercase n, lowercase italicized n. So that n, in the case of humans, that n stands for 23. So one complete set of chromosomes, the haploid number for humans is 23. So for humans, that is 23. Okay? 24 for chimpanzees, right? Now, when we double them up, right, we have one set of 23, an identical set of 23. Now we have 2 times 23, or 46, and we call that diploid or diploid. You can pronounce it either way. And we denote that with 2 times, an italicized 2 times lowercase n. So if you know the haploid number of any organism, you can always calculate very easily the diploid number by multiplying by 2. So for humans, 23 times 2 is 46. For chimpanzees, 24 times 2 is 48, like that, okay? So you want to keep those terms, haploid and diploid or diploid, and their designations, lowercase n, 2 times lowercase n, you know, kind of keep those compartmentalized in your own head. Another quick check here. Each chromosome is composed of 2 sister blanks joined together at a blank. Your choices are chromosomes centromere, so 2 sister chromosomes joined together at a centromere, 2 sister chromatids joined together at a centromere, 2 sister chromosomes joined together at a nucleosome, or 2 sister chromatids joined together at a nucleosome. So each chromosome, right, remember it's composed of chromatids, and they're joined together at a centromere. All right. Okay, what you see here is in this image, it's called a karyotype. And I'm not going to give you a lot of information here. It's really more just an interesting aside, I guess. But a karyotype is an image where your chromosomes are arranged in order from largest to smallest. There are 22, remember our n number is 23, there are 22, we call them autosomes, plus 1 pair of sex chromosomes. In this case you have the sex chromosomes determine male-femaleness. So you have an XX chromosome, that's female, and an XY, that's male. And you can see from this karyotype we have an X and the Y is little tiny. But you have an X and a Y, which tells you that this karyotype comes from a male. All right, now, and you can use a karyotype to determine chromosome complement or number, as well as any abnormalities. And actually, when I do this as a live lab, I have my students actually do a karyotype and then tell me, part of the assignment is to determine if there is a problem, or if this person is normal, or if this person is male versus female, or whatever like that. All right, just an interesting aside, really. Chromatin is a complex of DNA and protein. Chromatin, some people pronounce it chromatin, and that's okay. So it's a complex of DNA and protein. We'll break it apart and we'll see that in a minute. RNA is associated with chromosomes during RNA synthesis, but chromatin or chromatin is DNA, right? So it's a single chromosome is one long continuous double-stranded fiber, as I mentioned before. Now, a typical human chromosome, 140 million nucleotides. Remember, DNA is just a long strand of nucleotides. That's the monomer for DNA, is a nucleotide. And human chromosomes typically have up to 140 million nucleotides. I told you these things were like astronomically long. So in non-dividing nucleus, we say that the heterochromatin is not expressed, the euchromatin is expressed. Remember, EU, like eukaryote, right? EU means true, so true chromatin gets expressed. Heterochromatin is not expressed. So much of the information contained in your nucleotides, 140 million nucleotides long for one chromosome, does not get expressed. Only really a small portion of that gets expressed. Expressed means presented as a phenotype. Brown hair, green eyes, 6 fingers on your right foot, that kind of thing, okay? Now, when you unravel, and we will here in a second, but when you unravel chromatin, see, it's a complex of DNA and protein. When you unravel it, it looks like this. You can see these little circular structures. Those are your proteins, okay? Those are nucleosomes. And then where you see the little kind of strings between the little bumps, that's just DNA. So chromatin is DNA wrapped around these proteins. Now, the structure of a chromosome, okay, structure of a chromosome. It's a nucleosome is a complex of DNA and histone proteins. And I'll explain this the image here in a minute. Nucleosomes promote and guide the coiling of DNA. DNA is coiled around 8 histone proteins for every 200 nucleotides. Histones are positively charged and strongly attracted to negatively charged DNA. That's the phosphate groups. Remember, they have a negative charge on them. And without this kind of packaging, DNA cannot fit inside of your cells. Now, in my live classes, I have my live labs, I have my students extract their own DNA. Maybe you did this in high school biology with a strawberry or a banana. You can actually extract DNA from eukaryotic organisms. I always have my students extract their own DNA using cheek cells. They just scrape a few little cheek cells using a toothpick from the inside of their mouth, and they extract their own DNA. This is my DNA. This is just a little microcentrifuge tube, and you can see this little bit of white coiled up stuff. That's DNA. That's my DNA. Now, I did this for the first time like maybe 20 years ago, and I still have my little tube of DNA. And that's what this is a photograph of. But my point here in showing you this is that this DNA, which is perfectly visible, and before I put it in the tube, I could hold it in the palm of my hand, right? And I can look at it with the naked eye. That stuff was inside of my cells, which are microscopic. And the only way that can happen is if there's some sort of magical-like incredible packaging system of this DNA. And there is such a system, and that's what you see here. That's what we'll see. Okay, so these nucleosomes are wrapped in the higher order coils. We call them solenoids, and I'm going to show you an image here in a minute, or a diagram. And so when you look at a cell and it is not in the process of division, active dividing, that's what you see. We call the phase is interphase, and we call that interphase chromatin. So it's just this loose mass of chromatin. That's what you see. It's only when the cell is actively dividing the chromatin condenses, and that's when you see these X-shaped structures that we call chromosomes. Now, during mitosis, chromatin and solenoids are arranged around a scaffold of proteins to achieve maximum compaction, right, so that you can eventually see chromosomes. And we'll see the progression here in a second. So here you go. So if we begin here, this is just the loose double-stranded DNA, right, your double helix. Now if you take this DNA and you wrap it around histones, you're going to make these nucleosomes. See the histone proteins in the middle with the associated DNA wrapped around it. Then it looks like this micrographically. These little bumps or little circular structures on strands of DNA, we refer to that as the beads on a string presentation, okay? Because it kind of looks like, well, beads on a string. So what this is, however, are nucleosomes connected together. Nucleosomes are histone proteins with their associated DNA. So we're getting more compact, right, as we're going through this illustration. From loose DNA to beads on a string to nucleosome, we're just looking at a detail of nucleosomes, and the nucleosomes bundle together into solenoids, and these scaffold proteins here then take these solenoids and they loop them together. So we see a chromatin loop, and these loops are joined together, right? So we see these, they call them rosettes because it kind of looks like a little flower here. So we're getting more and more and more condensed, right? We're going into mitosis, so we need to make chromosomes that we can see with a light microscope. So we're getting more and more condensed as we go, as we follow the illustration here. We're actually going backwards here, but so then you get these rosettes and they condense even further, and then you end up with a chromosome. This is a mitotic chromosome. This is a chromosome ready for cell division with your two sister chromatids joined together at a centromere. These are kinetochores, these little green things, and we'll get to that in a little bit. All right, so if we break it down, if we go in reverse, here's your chromosome, we unravel it, we get the rosettes, we unravel them, we can see the individual chromatin loops, we unravel them, we see solenoids, we unravel solenoids, we get beads on a string, we unravel the beads on the string, and we get the single-stranded DNA, or the double single strand, one long single strand of double-stranded DNA. Okay, time for a quick check. Histone and its associated DNA is better known as a solenoid, nucleotide, chromosome, nucleosome. If you answered nucleosome, you're absolutely correct. Remember, you have histone proteins with its associated DNA wrapped around it. That structure is called a nucleosome, and a bunch of nucleosomes together is then called a solenoid. So, chromosome replication, here we go. Replication is duplication of chromosomes. It occurs prior to cell division. We have to make another copy of our chromosomes before we divide them into two halves so that each daughter gets a full complement of chromosomes. The process is called replication. So what happens? Each chromosome is copied, and the copies, sister chromatids, are held together at the centromere. So we know that already. So here are your sister chromatids held together at the centromere. So here's where it kind of gets a little bit fuzzy sometimes for students. Okay, so here you see two chromatids, right? These are chromatids from different chromosomes, from homologous chromosomes, right? Now, at this stage over here, sorry to jump back and forth, but at this stage over here, these are homologous chromosomes, and each homologue is composed of two sister chromatids joined at a centromere. When you pop these chromatids apart, okay, you pull them apart, they're no longer connected. Now, because each chromatid has the full genetic complement of the chromosome, now we refer to them not as chromatids but as chromosomes. This will make more sense, I think, when you see the actual process, because at one point in the cell's history, we have 96 chromosomes, 92 chromosomes, way more than the 46, right? So when the chromatids are connected together, and by the way, these sister chromatids are genetically identical, right? And these homologous chromosomes are genetically identical, right? And so when they're connected, it's a chromosome. When they're connected, these are chromatids connected at the centromere to form a chromosome. When you pop them apart, now each one becomes its own chromosome because it has a full genetic complement as the chromosome over here, okay? Tuck that in the back of your mind. It'll come up again. All right, so mitosis is a process of cell division, separates sister chromatids so that each new daughter cell receives one complete set of 46 chromosomes. We'll look at the process. I think it'll make more sense. Okay, so here's just a kitschy way of presenting mitosis to you. This little video, it's called the mitosis song. I only put it in there for comic relief. All right, if you have any questions, message me 804-229-7993, email jjeffries@rbc.edu, and let me know what's going on, okay? That's the end of the first lecture. See you in the next round.